Hey, what's up, guys? Happy Friday. Made it to the end of the week here. And man, it's been a long week. But it's really good to see you guys yesterday at the live stream. It was I ended up doing three more after that with my freshmen, and they're hilarious. And it was just it's weird. I was telling my wife after that, even though we're not in the same room, like it's funny. Even like the live streams, each live stream like felt different, just because every class is different. So it's part of the reason. One of the reasons I love this job. It's just you guys keep it really interesting. Anyways, hope that was really helpful and fun for you guys. We actually have our next live stream on Tuesday at 9:30 in the morning, and that'll give us a chance to kind of recap both this chapter, which is chapter 11, and the one we're going to do on Monday, which is the beginning of the neuroscience stuff, which is super fun and really cool to talk about. So I'm going to get right into it because you didn't turn this on to listen to me go on and on and on. We're going to switch over to our lesson for today, which is chapter 11. This is looking at cell communication. And you may be wondering, like, why are we going all the way back to chapter 11? We made it all the way into the 40s, and now we're going backwards a little bit. But I've kind of been saving this chapter because in years past, we would teach this at the beginning of the year, with all the other like cell stuff, like parts of the cell, membranes, and all that kind of stuff. And by the time we needed those concepts, we really didn't use any of this stuff until more recently with the immune system. So it kind of just made more sense to bring this chapter to the physiology unit, just because there's a lot more application. So that's why we're doing this. And after you watch this, you're going to be working on the chapter 11 reading guide and getting a little more review of everything we're talking about. So let's go ahead and get started. So as you've kind of seen by now, even just with the immune system, in order for the immune system to work, it really relies on organisms being able to have cell-to-cell communication. I've said this many times, but cells don't have senses like we do, right? And so the way that they know what's going on around them, or the who's around them, or what's around them, is using cell-to-cell communication. And really what that comes down to is using chemicals. And so it's super important, not just for animals, but all organisms, that they're able to communicate with one another. And as we kind of evolve towards being more multicellular, it becomes even more essential. Why do we need it? Because that's needed for reproduction. Even like something as simple as a sperm and egg, we look at it and we're like, that's a sperm, that's an egg, they go together. But if you look at it from a cellular perspective, a sperm is just a cell, and an egg is just another cell. So for them to come together and combine their chromosomes, there's a good deal of communication that needs to happen there. Responding to environmental changes, right? The conditions, whether bacteria or human, are always changing around us. We have to be able to sort of sense those changes and respond. And then, as we saw in the immune system, cell-to-cell recognition is really important for distinguishing between your own cells and the cells that are not supposed to be there. So there's I kind of skipped ahead a little bit too quickly, but there are several different scales at which communication can happen, okay? And what we're going to do is kind of start with the closest one, which literally is just touching, that's direct contact, and then we're going to back up and look at how communication happens within our body over longer and longer distances. So with direct contact, what that means is literally you're so close, these two cells are so close that they're actually sharing their cytoplasm with one another. And before I show you the words, can you guys think just in your head, what structures need to be present in the cell for the cytoplasm to be shared? I'll give you a hint, plants have a version of it, and animals also have a version of it too. Give you a sec to think about that. You got it? All right, let's take a look at it. So in plants, we have gaps in the cell wall, as well as the cell membrane, that allow for not only cytoplasm to move back and forth, but also nutrients and hormones that are really important for communicating. And what do we call those? Plasmodesmata. Hopefully you got that. In animals, we have something similar. We don't have a cell wall, but there are these channels or gaps in our membrane called gap junctions, and that again lets cells come in direct contact with each other. So if you have cells that are this close to one another, they still need to be able to communicate. If one thing's happening in one cell, the other's going to want to know what's going on there. So they're going to be sending those chemicals back and forth to each other. Another form of direct contact is something that we saw in the previous chapter with the immune system, right? And this actually, I know some of you guys might look at that and you're like trying to figure out, is that a B cell or a T cell or MHC or whatever? And actually, this picture doesn't show specifically an immune response, an immune cell. This is just showing how one cell can communicate with another by binding to that receptor. And our immune system cells do have that, but it's not just our B cells and T cells that have this kind of like receptor structure in their membrane, and then they come in contact with one another like that. So it's not just unique to your immune system, but that's going to be really important for cell-to-cell recognition, right? This is the mechanism that we figure out whether or not those cells belong there. And so what we're really talking about here is an interaction between the membranes of the cell surfaces. I don't have the picture here, but you guys probably remember from the very beginning of the year that you have these intermembrane proteins that are stuck within the lipid bilayer and facilitate all kinds of things like active and passive transport. But as you can see here, many of those proteins are also going to act as receptors pointing towards the outer world, right, outside of the membrane, to receive messages and to talk to other cells. The immune system is a great example of that. Now, the next level, kind of going a little bit further, is local signaling. And this is where cells are nearby each other, but they are not actually touching, right? And I guess to kind of tie it in, just because the theme right now is all about coronavirus, and the coronavirus analogy, the direct contact, right, that we see from the previous slide, that's like you go and you touch someone that's sick and you make out with them and you get sick. The next slide here, local signaling, is kind of like you take a few steps back, but you're still within range of contracting that disease, right, or receiving those chemicals. Now, this obviously is not going to be for disease. Local signaling is really where a cell is going to secrete some kind of messenger molecule, and then it doesn't really travel that far. It just kind of floats through the interstitial fluid or the extracellular fluid, and as it diffuses, then those chemicals are going to bump into any cells, the target cells that are nearby. Okay, and so there's kind of two types that we see here in our body, at least. One is called paracrine signaling, and that's what this picture right here shows. I'll go ahead and zoom in on that. In paracrine signaling, you can see this is the secreting cell, and inside it's producing some kind of regulator or chemical messenger. Okay, we don't really need to worry about what it is. If you need an example, mast cells would be an example of that. Right? We talked about that in the immune system. Mast cells would have vesicles filled with histamine, and then when your cells are damaged, they're going to release that histamine to the local area, and that's going to trigger vasodilation and the inflammatory response. So that would be an example of paracrine signaling. But you can see here, when the chemicals are released, the target cells are going to be receiving that message. And what they left out of this picture, now that I look at it, is they don't have any way to receive that, right? So in order to receive the message, they would have to have some kind of receptor that fit perfectly with these little red dots, and then that would trigger some kind of response from the target cell. So that's paracrine signaling, where the signal is just released into the extracellular fluid. Now, another version, which is kind of similar, but it's kind of in a different context, is synaptic signaling. And in the same way, chemicals are being released into the extracellular fluid. So why do we just call this paracrine signaling? Because this is specific to neurons, which we're going to start talking about on Monday. If you're kind of familiar with neurons, you'll know that they don't actually touch, which is pretty incredible. You can send a message from your brain down to your foot through, just maybe hundreds of neurons are passing that message from one another, but they're not actually touching each other. There's kind of a gap here called a synapse. And so when one neuron, what we call the presynaptic neuron, is sending the message and wants to pass it on to the next neuron, which we call the postsynaptic neuron, it'll, in the same way, bring vesicles that have these little chemicals. In neuroscience terms, this is called a neurotransmitter, and it'll dump that neurotransmitter into the synaptic gap, and then on the postsynaptic neuron, the one that's receiving the message, they would, again, it's missing from the picture, but they would have receptors that the neurotransmitter would bind to, and then that would trigger a new message to be sent out this neuron. And so when you're doing something as simple as like moving your finger like this, all the neurons from your brain to your finger and back are releasing this message, and then that's kind of playing a game of telephone using these chemical signals. So that's synaptic signaling. And again, these are both examples of local signaling because these chemicals aren't really meant or expected to travel long distances. It's really just covering that tiny little gap between one cell to the next. Okay, now let's go long distance, right? When we're talking long distance, now we're saying like it literally can be from several inches to several feet. Okay, the one I can think of like that comes to the top of my head is with the reproductive organs, right? There's signaling, there's communication that happens from your pituitary gland, which is like right by your brain, and it actually sends a chemical message all the way down to your genitals, to your testes and the ovaries to communicate and regulate the whole process of puberty and also reproduction. So I don't know how tall you are, but you could try to measure the distance between your brain and your genitalia. That's a weird way to say it. Your brain and your gonads is the proper word. Yeah, gonads, ovaries and testes. It's where your gametes are made. So if you think about that distance right there, right? I don't know, what is that, like 3 feet or something? You're actually like literally sending chemical messages over 3 feet, which on a molecular scale is huge. That's super, super far. So how to ensure that the message is going to get there? Well, if you haven't already noticed in the picture here, it's going to hop and catch a ride in our transport system, which is our bloodstream, right? That bloodstream that's already being used to carry oxygen and nutrients and all the things we talked about in the previous chapters. It's also going to be a highway for hormones to get dumped into, and those hormones then can get carried over long distances. Now, I use that word, this is the first time I've actually used that word hormone. So you want to know hormones are chemical messengers that are going to be carried through the bloodstream. Okay, and so as I mentioned, it's going to rely on a transport system, which is going to be your circulatory system, but it can also be that other system, that lymphatic system. That's that set of tubes that your white blood cells are kind of using to move around between your lymph. Hormones can also travel through that. Okay, now the interesting thing about this is that because it's going in the bloodstream, every part of your body is actually coming in contact with the hormone, right? So when you're going through puberty, for example, your brain releases a hormone called GnRH, gonadotropin releasing hormone, and that's released from the brain, and that triggers a release of other hormones like LH and FSH, right? Luteinizing and follicle stimulating hormone. We'll talk about that in another chapter. But those hormones then travel through your bloodstream, and the target for that is the testes and ovaries, right? Which is 3 feet straight down. However, because it's going in your bloodstream, and by the nature of your blood, your circulatory network, all the cells in your body are going to be exposed to LH and FSH. And so the question is, are all the cells going to respond? Of course not, right? If the instructions are just like we're releasing this to tell the testes to make sperm, or telling the eggs to produce telling the ovaries to produce eggs, that message isn't going to be useful in your hand, right, or in your stomach, or in your ear. So there's a mechanism then that allows these hormones to only affect targeted cells, and that is that the target cells are the ones that have the receptor. Does that make sense? So even though all the cells are watching this hormone float by, only the cells that have a receptor are going to actually bind to that hormone, and then that's how they actually get the message. Okay, and so it's kind of like this code, this message that's like broadcasted to everybody, but you kind of have to have a way to tune in to get that message in order to be affected by it, right? And that's how, even though the whole body is exposed to it, you can get a really specific response. Now, how is that possible? Because specificity is all about receptor shape, right? Just like the active site of an enzyme is specific to its substrate, these receptors are also specific to the chemical messenger that is going to bind to it and then activate it. So that's how long distance signaling happens. Now, what happens when you actually bind to it is we go and through something called signal transduction. And this should be reviewed for us by now because we talked about this in the plant unit. In signal transduction, let's see, picture is not coming up. Oh, here we go. I'll just kind of show you the picture right here. So in signal transduction, it's basically the process of receiving a message at the cell membrane, right? So like kind of the molecules like knocking on the door and saying, hey, I'm here to carry a message. And the first thing that's going to happen, as you guys already learned, is that we have the process of reception. In order to receive that message, you have to have a receptor there. No receptor, that means the molecule floats by and you don't get a response. The second step is transduction, and that's where this purple protein here is going to relay that message to what's called a signal transduction pathway. Right? It's a series of proteins where one activates another, that activates another, and passes that message on. Now, ultimately, where do we want to get that message? Well, you want to get it to the part of the cell that is going to control everything. And so, what's the kind of the big boss of the cell? It's going to be the nucleus, right? So one way or another, what's going to happen is you're going to activate a cellular response, and that usually involves some level of transcription or translation at the nucleus, right? If you think about it, whether the response is to go into mitosis or apoptosis or to secrete a certain protein or make enzymes, it always comes back down to making some kind of protein. So chances are that message is going to find its way to the nucleus and then turn on some kind of transcription factor. Okay, so again, you guys already kind of have learned about this, but I want you to kind of connect this concept of signal transduction with all the examples that we've talked about before, right? Whether it's, I'm going to go backwards here, long distance signaling, whether it's local signaling, or even direct contact. Anytime you have some kind of message arrive at the receptor, you can see even they drew arrows right here to represent that the message now is being sent into the inner parts of the cell through a signal transduction pathway. So that this picture right here of signal transduction, that's a very common theme in all the different ways that we communicate in our bodies. Okay, just a little more about receptions. There's kind of more than we need to know here. I'm really going to focus on this part right here. Okay, I've already kind of discussed how the signal molecule is a complementary fit. But something I want to focus on is the location of the receptors right here. You can see that there's kind of two groups or categories here. One is in the plasma membrane, and one is intracellular, which means it's on the inside. Okay, and these are the two places where receptors can be found. Now, if you look at the first picture that I showed you, this one right here, question: what kind of receptor is this? Okay, is it the plasma membrane or intracellular? Well, obviously, looking at the picture, that's in the plasma membrane. So something to think about here is this. If the receptor's in the plasma membrane, that means that the signal molecule is not able to get through the lipid bilayer on its own. Okay, and just to tie in something from previous topics, what does that tell you about the chemical properties of the signal molecule? I'll give you a sec to think about that. Okay, hopefully you're kind of thinking the signal molecule can't pass through the lipid bilayer. That probably means it's impermeable. And why would it be impermeable? Because one, it's either too big, or most likely two, it is polar. Okay, so if that signal molecule has any kind of polarity, it's not going to be able to get through that hydrophobic core of the lipid bilayer. And for that reason, it has to bind to a receptor that's out here in the membrane. Okay, now the opposite of polar is nonpolar. And if it's nonpolar, that means it can get through the membrane, right? So are there chemical messengers that can get through the membrane? Absolutely. Great example of that in this picture would be testosterone, which happens to be a type of steroid. Okay, and you guys may remember steroids fall in the category of the lipids and fats group, right? So they have those hydrocarbon chains, even though they're folded up into rings, they're very hydrophobic, which means steroids like testosterone, and estrogen, progesterone, they can just diffuse directly through the membrane. And that makes them really, really powerful, because that means that there aren't as many ways for your cell to regulate whether that message is coming in or not. It's just going to help itself through that front door and come in. Now, the hormone on its own can't just go straight into the nucleus and like bind to your DNA. Okay, at some point, it still has to bind to a receptor. In this particular picture, the receptor protein is a protein that's already in the cytoplasm of the cell. And in the same way as you saw with the plasma membrane, it's going to bind to that receptor, and the receptor then goes to the nucleus and triggers transcription and translation of some kind of new protein. Okay, so that's what this whole piece here is really saying, is that in the plasma membrane you have hydrophilic, otherwise known as polar signal molecules that will bind to the receptor, whereas in the intracellular ones, they tend to be hydrophobic, directly crossing into that membrane, and they can directly activate some kind of hormone receptor complex that then turns on certain genes. Okay. Now you see some other things here. It says 3 types of plasma membrane proteins or receptors: G proteins, tyrosine kinases, and ion channels. We are going to, I'm kind of off the screen here. We are going to talk about those in the following slides. Okay, but before we get in that detail, you do want to remember that that there's these 2 different categories of reception. Okay, so let's take a look at those 3. The first is called a G protein linked receptor, and sorry, I keep trying to grab my, where it is, I'm trying to grab my camera, but it grabs the whole slide. So a G protein gets its name, I think, because it actually interacts with GTP, which is a cousin of ATP. And there's not really any text. I don't think there's much text in these next couple of slides. They're just pictures. So try to follow along, and if you need to at any point, just kind of pause it and kind of take in the pictures. These are also straight from your textbook. So structurally, G proteins, again, they sit inside that lipid bilayer because they have this whole hydrophobic core that allows them to sit within that hydrophobic region of the lipid bilayer. Okay. Now, on the outside of the cell, as well as inside of the cell, there are ribbons and parts of that protein that are going to act as binding sites, as well as parts that interact with the inner portion of that cell. So this is kind of like a side view, a close-up of what that G protein looks like. Now zoom out, and this is what we have here. Okay, you have the G protein, you got that binding site where the signal molecule is going to bind to. Now, in addition to that, the setup that they kind of have here for you to see how this works is we have this enzyme over here. Okay, and this enzyme right now is inactive. It wants to catalyze some reaction, but it can't because it's not in the correct physical conformation. And so what we need to do is to change that shape so that it can carry out that reaction. So how do we do that? We're going to send a message from the G protein to the enzyme. And the middleman for that is this thing called a G protein. Okay, and so this is the G protein linked receptor, and this is the actual G protein. Okay, so this receptor is linked to the G protein. The G protein you can see right now is labeled as inactive. Let me zoom in on that so you guys can see it a little bit better. It's inactive right now. What we're going to do is when we activate it, it's going to go over to the right and activate that enzyme. So let's see how it works. When a signal molecule that is hydrophilic binds to the receptor becomes activated. And the G protein will actually take a GTP and bind to that G protein, which kind of provides the energy to activate that protein. Now you see this GDP leaving. Remember, ATP, when it gets used, turns into ADP, right? Well, GTP does the same thing. When it gets used, it turns into a GDP. So this GDP is actually sitting there from the previous time that this G protein was used. Okay, so this is not giving any energy. When the receptor is activated, a second GTP will come in, kick out the GDP, and bind to that same spot, and then it provides the energy to run that communication. Okay. Now, once that G protein is activated by the receptor, then the GTP will then the G protein with the GTP on it will travel over to that inactive enzyme, activating it, and then that enzyme does some kind of chemical reaction, which then causes a chain reaction that ultimately leads to some kind of cellular response. And now we've achieved our goal. Okay, and you can see over here the receptor, having done its job, is going to release that chemical messenger, and it's going to float away, and the message is basically cut off at that point. Okay, now once that happens, we're going to go back kind of to a resting state. We're going to try to restore it back to what we had at the beginning, and so you can see here the phosphate of the GTP is going to be has lost its energy right now. It's lowered in its potential energy, so it breaks off, and now that GDP is left behind on the inactive G protein, and that's the same. That's how and why we started with that GDP up here in the front, and now it's kind of waiting for another chemical messenger to come in. Okay, so that is like a pretty specific way that we can bind a signal molecule and then trigger a cellular response. Now, the next kind is also kind of similar, but it's a little bit more complex just because there's more moving parts to it. This is called a tyrosine kinase, and these always show up in twos. They're monomers. Each of these is a monomer, and they get their name because of these tyrosine amino acids on the side. Okay, you don't need to worry about that too much. So, how do we activate this, and what happens when they get activated? Well, here comes our signal molecule, right? And where do you think it's going to bind? Well, it's kind of circular shaped, and you can see that we have a signal binding site right here and right here. So when you have two signal molecules come in, each one's going to bind to each of the tyrosine kinase monomers. And when the two monomers come together, what do you get? You get a dimer, okay? And now that dimer is activated and can transmit some kind of information. By the way, the word kinase, we've talked about this before. If you remember way back to mitosis, we talked about, what was it called? Cyclin-dependent kinases, also called CDK. A kinase is really just a protein that acts as an enzyme, but its specific job is to turn on other proteins or other enzymes. Okay. In other words, the job of this tyrosine kinase is when it gets activated by these signal molecules, their job is to now turn on other proteins and start that chain reaction of communication within the cell. And so here you can see it doing that, right? Now, to do that, it needs to get energy. And where does it get energy from? This time it uses 6 ATPs. That's a lot of energy. Those 6 ATPs will come in. They will phosphorylate the tyrosine kinase regions. In other words, a phosphate is taken off of the ATP and it gets stuck onto that tyrosine. And you can see here now all 6 tyrosines are fully activated and you have a phosphorylated dimer. Okay, now it's ready to actually send the message. And so you can see here that you have some inactive relay proteins, right? Their job is to pass the message on to other proteins. Well, they're inactive right now. But when this tyrosine kinase gets activated and by the molecules and the ATP is phosphorylating these kinase enzymes, that's going to cause these relay proteins to rush over here, bind to it. They're going to get the energy from the phosphate, and then they will activate different cellular responses. And in this case, this first blob here is going to activate cell response 1, and the other one's going to activate cellular response 2. Okay, so that basically turns on the cell to say, hey, cell, we got the message, now respond in a certain way. Do mitosis, make more enzymes, whatever it is. Okay, so that is how a tyrosine kinase works. The third and last kind of plasma membrane receptor is a ion channel receptor. And this is going to look really familiar to you guys because we did talk about ion channels at the beginning of the year, right? Remember ion channels, before we get to any of the regulation stuff, are simply proteins that function in transport. Now, question for you to think about, is this an example of passive or active transport? If you thought active transport, you are wrong. It is passive transport, right? Remember, channels do not actually move anything against the concentration gradient. All they do is they open up and they allow solutes to move from the side of higher concentration to the side of lower concentration. So this is completely passive, okay? Now, even though it's passive, it's highly regulated. And especially when we get to the neuron on Monday, we're going to see that ion channels are actually the thing that's responsible for regulating neurons firing these signals, okay? And that's what allows you to have thoughts and have memories and recognize people's faces and have emotions and all of that stuff. So all that's happening because these channels are opening. Now, in order to open at the right time, we have to regulate it. And that's where you have ion channels that are gated, okay? And so a gated ion channel gets its name because it has this little door that has the ability to close, and that prevents the ions. In this case, the ions are the solute that's going to move through. They are not going to be able to move through if that gate's closed. Okay. So how do we get this this gate to open? Well, you can use a signal molecule, right? If you have a ligand-gated ion channel, there's the key term for you right there that you may want to write down. A ligand, to ligate means to glue something together. So a ligand-gated means that a signal molecule, also referred to as a ligand, is going to bind to that specific site. And when it does that, the gate's going to open and then allow the ions to move down the concentration gradient. And in this particular picture, the gradient favors the movement of ions from the outside of the cell, the extracellular fluid, into the cytoplasm. And that's exactly what happens here. Now, once that happens, those ions can trigger some kind of cellular response. Okay, now we don't need to worry about the details of that right now, but that cellular response in the neuron could be the firing of a new, what we call action potential. That's a nerve impulse, basically, can be generated. If this is in the muscle, the cellular response can be a contraction of that muscle. So actually, I mean, you don't even realize this, but depending on what muscles or what setup you're looking at, when these channels open, that's what actually causes your muscles to contract. Okay, so and we'll look at that next week when we look at the muscular system. Now, again, this is a reversible process. So, you know, as long as there's a concentration gradient, as long as that ion channel is open, the ions are going to keep moving, right? But because this is a regulated process, as soon as that ligand is removed from that binding site, the gate's going to close, and even if the gradient's there, those ions are not going to be able to move through. Okay, so those three things, the ion channels, the tyrosine kinases, and the G protein receptor, G protein linked receptors, those are all examples of the plasma membrane receptors that receive messages from hydrophilic, otherwise known as water-soluble signal molecules. Whereas the intracellular ones, we said they're hydrophobic. Another word for that is fat-soluble, which just means that they can dissolve within fats, like your membrane. Okay, so let me click through all of this now. A couple of things to kind of close with. I think we're getting close to being done. Transduction always involves a number of steps here, right? You got to think about this kind of like a game of telephone. You know, you don't just have one person talk to another person. You have like 20 people in a row, and they're all passing this message one from another to another to another, and so on. And so transduction has a lot of steps, which is a really good thing because it provides us with a couple opportunities. Number one is actually provides you with an opportunity to amplify that signal. Okay, what I mean by that is if you think about I'm not going to show you the picture that we just looked at, but I was checking to see if we had a picture on this slide. We don't. But if a signal molecule binds to a G protein linked receptor, right? That is like one molecule binding to one receptor. And if you only had a couple steps there, you know, that one receptor might only activate one protein, which would activate another protein, and then that gets to the nucleus, and you just have like this kind of weak message of saying like, hey, this one message came in. The way amplification works, though, in real life is that, I'm trying to see where the picture goes. I guess I have to show you all of it. There it is. Okay, that's the picture I want to show you. Is that even though one molecule can bind to an active G protein, the number of G proteins that get activated as a result of that can actually amplify. So in other words, the receptor, instead of just activating one G protein, you know, that inactive one with the GDP attached to it that migrates over, instead of it activating just one, when that receptor is activated, it can actually activate 100 G proteins all at once. Okay, which means that you just went from one signal molecule to 100 G proteins that are now activated. And then those G proteins that are activated will then go and activate other enzymes, and they can activate multiple enzymes as well. So in this picture here, you can see it starts off as one molecule, and then it activates 10 to the second molecules of G protein. They then activate 10 to the second molecules of this adenylyl cyclase enzyme, which then causes the cyclic AMP to rise by 10 to the fourth power. You don't need to memorize all these. But you can see as we go through, every single step of the way, the scale of all of this messaging increases. So by the time you get to the cellular response, you've now are 10 to the 8 molecules of this glucose 1-phosphate. Okay. Now, what is all this showing? I think since this is epinephrine, this is showing how one molecule of epinephrine can tell your liver to break down glycogen into glucose. Okay, so you know, if we didn't have amplification, what that would mean is one molecule of receptor would lead to only one molecule of glucose being released, which is not very efficient, right? When you're having if you didn't know, epinephrine is also known as adrenaline, but when you're having an adrenaline rush, you're trying to you're trying to pump out as much sugar as you can into your bloodstream so you can have that fight or flight response. So if you wanted to release 1 million molecules of glucose and it was a 1-to-1 ratio, you would have to release 1 million molecules of epinephrine. And that's a lot to ask for. But what you can see here is that one molecule of epinephrine can lead to 10 to the 8, which is, 10, 100 million molecules of glucose. Okay, so that's the amplification that we're talking about here. So the signal can be, oops, sorry, too much. Oh gosh, sorry, it's been a long day. Where were we? There we are. Okay, so the signal can be amplified, and that produces a very, very large cellular response. And then also, in addition to that, kind of separate from the amplification, when you have all these steps that are involved, that provides more opportunities for coordination and regulation, right? So if you start this cascade of messaging, but then we decide, oh, we don't want that anymore. Every single one of these steps represents a place where the cell can step in and say, okay, no more, or it can step in and say, actually do it even more. Okay, so that's why transduction has so many different steps to it. I'm going to skip over this part. Don't worry about that. Let's see. It is kind of important to know within the cell that not all members of this transduction pathway have to be proteins. Okay, in the examples we looked at so far, there were G proteins and enzyme proteins and so on, but you also have non-protein relay molecules that are made in very high quantities and can spread through the cell very quickly by diffusion, and they don't really need the phosphorylation or the activation that a protein might need. Now, some examples of that, we've actually talked about some of these before. That includes cyclic AMP, right? Or otherwise known as CAMP. And if you remember, CAMP was the one that binds to the cyclin-dependent kinase, the CDKs, to make MPF. That's the one that during the cell cycle, you guys remember, the amount of MPF goes up as we get closer to mitosis, and then drops down, and then goes back up, and then drops down. Well, remember, all that was regulated by the levels of CAMP, or cyclic AMP. So that's an internal secondary messenger inside the cell. Okay. Another example, as we'll see next week, is calcium. That's an ion, and Ca2+, and that's going to be really helpful to trigger release of certain things, from the cell membrane to the mitochondria, as well as the endoplasmic reticulum. So just know CAMP and calcium as secondary messengers. Even though they're not actually proteins, they have the ability to turn on other functions within the cell. Okay. So here's kind of what I want to end with is just a diagram that you've never seen before. This is a good skill for the AP test. Looking at a diagram you've never seen before. So here's kind of my challenge to you, okay? And this is going to involve you actually pausing your video just so that you have some time to look at this. What I'd like you to do is to pause the video and then create a narrative, a storyline of how we get from the signal molecule here to the release of calcium. Okay, this picture doesn't show it, but the goal here is to have this calcium that is stored inside the ER, the endoplasmic reticulum. We want that calcium to move from the ER out here to the cytoplasm. Okay, why do we do that? You don't need to worry about it right now, but we just want to get these green dots out of this area. Also notice there's no calcium out here, so there's a bit of a concentration gradient. So the calcium already wants to get out there, it just needs a way to get there. So I'm going to ask you guys to go ahead and pause and walk through. Oh, sorry. Shoot, I gave way too much. That's what we want it to happen. We want the calcium to go out and then trigger some kind of response. Okay. So before we get to the response, oh my goodness, I'm really struggling with this. I'm just gonna. I think the scrolling is just messing with it. Okay. So go ahead and pause it right now. Start with the signal molecule. Walk through all the things you're not going to know what PIP2 and DAG are and IP3. Don't worry about that at all. Just treat them as as messengers in this transduction pathway. Okay, so go ahead and pause right now and try to figure out what's going on here. Okay, so you've paused it, you've worked through it. Let's see if you've kind of figured it out. Here's kind of the first step. And by the way, this would be a great essay question if this shows up. So you maybe want to know it well enough that you can write it out. Step one is the signal molecule here. This red thing is the ligand. That's going to bind to this G protein linked receptor in the plasma membrane. Right? That also tells us this molecule is water soluble, that it's hydrophilic, it can't pass through the lipid bilayer. That G protein linked receptor gets activated. It then activates the G protein, which takes in that GTP that gives it that that energy to wake it up. And now the G protein has the ability to activate another protein, which in this case is this guy right here, which is phospholipase C. Okay. Now phospholipase C has a job, which is to come over here to this thing hanging out in the lipid bilayer. Now, you've never seen this before, but just kind of use your I don't know what the right word for this is, but just visually look at it and look at the differences, right, between this and these two things. Hopefully what you notice is this is basically just this molecule called DAG, and IP3. If you connect them, that's what you get. That's this molecule here called PIP2. Okay, so you can see the action here is that PIP2 is going to be broken. We're going to cut that bond right here and leave the DAG portion in the membrane, and the IP3 is going to float down as a secondary messenger into the cytoplasm. Now, what is going to cause PIP2 to disassociate and to break those two things apart? That's the job of phospholipase C, okay, which again is activated by the G protein, which is activated by the G protein linked receptor, which is activated by the signal molecule. Okay, it's like a whole process right here. Now, once IP3 is made, what is its job? Well, IP3 is going to move over here to the ER because remember, the goal is to release those poor calciums that are stored up inside the ER. And notice they're being blocked right now by what? An IP3 gated calcium channel, which means that channel is only going to open up if an IP3 binds to it. Now, where do we get the IP3? From the PIP2 breaking up here. Okay, so hopefully what you figured out was the IP3 travels over here, binds to the IP3 gated calcium channel, and then opens up that channel, and now the calcium is released into the cytoplasm. And because calcium is a secondary messenger, that's going to then, and I know they're kind of shortcutting here, that's going to activate other proteins, which activate other proteins, and ultimately you're going to get some kind of cellular response. Okay, now this is kind of where the multi-step thing we just talked about comes into play, right? Some of you guys might be thinking like, oh my God, that's so much work. Why don't we just have a signal molecule bind to the G protein and have the G protein cause a cellular response? And again, the reason you don't want to do that is because when you have multiple steps, that allows for more regulation, where we can intercept this whole process. And even though the picture doesn't show it, there's amplification happening too. Okay, remember this one G protein linked receptor is not activating just one G protein, it could be activating hundreds, which then could be activating hundreds of phospholipase Cs, and that's going to release potentially thousands of IP3s and open all kinds of channels throughout the cell, and the calciums here are going to be very, very amplified. Okay, so that's why it's beneficial to have like those multiple steps. Okay, even though it seems easier just to skip steps. So I hope that makes sense. If that doesn't really click with you, maybe put a star next to your notes, and we can go over it during the live stream on Tuesday. Okay. I'm gonna skip through all this stuff here and get to the last part, which is just to remind you that when the signals bind to a receptor, it doesn't stay there forever. It is a reversible process. Sorry, let me move the words up. My face out of there. And so once the receptor is free, then everything that was active now reverts back to being inactive. Okay, so the last thing is why does this matter so much to animal physiology? We're in this physiology unit. Why did I save this up for now and not do this earlier? Because one, we are seeing these kinds of communications taking place all over the place, right? Whether it's direct contact, local signaling, or long distance signaling, all of those are going to utilize these surface receptors, these ligands. They're going to use G proteins and tyrosine kinases to activate those cells. And where do we see that taking place? Well, there's a number of places. We've already seen it in the immune system. It's critical for these cells to be able to communicate with each other and recognize MHC antigens and even cytokines, right? Remember the cytokines that are released by the helper T cells? Those cytokines are actually going to be released, and they bind to receptors on the B cells and the cytotoxic T cells, and that will activate them. That would be, probably some kind of mix between local and long distance signaling. And then in the future chapters to come, we're going to be looking at the nervous system, which is going to be using neurotransmitters to send messages, and that's going to be that local signaling. And then, of course, when we get to the endocrine system, towards the end of all this, we're going to look at all the hormones that are used to communicate with our body, and that's going to regulate everything from puberty to homeostasis to we have already talked about ADH hormone, which regulates your kidneys and water balance and all that stuff. All of that is made possible by cell-to-cell communication. Okay, and so that's why this stuff is so important. And 45 minutes went by already. That's crazy. All right, I hope that was helpful for you. And just a little tip: there is on YouTube down here somewhere. If I'm going too slowly for you, you can just play at 1.5x. See if that's manageable for you. All right, so get to work on that reading guide, and if you have any questions, feel free, actually before even Tuesday, if you have questions, just post them below in the comments from the reading guide, and I can help you with that. And if I don't hear from you, then have a great weekend. I wish that meant we could go out. I haven't even thought about it yet, but I'm definitely going to pull back from some of this work and actually rest up a little bit this weekend on Saturday and Sunday. And then we'll be back at it on Monday. All right. So have a great weekend, guys. Take care. Be safe out there. I will see you later.